There we go. So welcome everybody. My name is Beth Foss and thank you for joining us on a Friday. I'm super excited that you're here today, and we're going to be starting a new webinar series that those of us in the staffing are really super excited about. The board of directors is really making an effort hard at using your money wisely. And so we are funding some extraordinary researchers, and Dr. Fahim is starting us off. So we'll introduce her in a second, but I just wanted to quickly go over again that this is going to be recorded. It will be on our website. It will be on our YouTube channel. So please look at it, share it. We're going to use the same old ways that we've always done. I'll be running the chat line. So if you have questions and it's a reasonable time, I'll jump in and ask her at the time. Otherwise, we'll wait to the end. As always, we always want to say thank you to our sponsors, and our sponsors are Genentech, Santen, Biogen, and Spark. It's a mouthful, so I get those mixed up sometimes. So as I said, welcome to Dr. Fahim, Abby Fahim. She comes to us from the Kellogg Eye Center, and that is in the University of Michigan, Ann Arbor. And she was awarded a grant from the CRF and CRF Canada in 2020 to take a look at induced pluripotent stem cells, iPSC, and deriving them from pigment epithelium. So that is about all I know, and I'm super excited to learn more as she's going to share with us what the importance of this research is for some foundational aspects of choroideremia. And we're really excited to have you here. Thank you very much for taking
Thank you so much.
Time out of your schedule.
Thank you very much. This is a great opportunity to talk to everyone about this. I'm going to share my screen. Hold on just a second. Let me just get that adjusted there. Okay, good. Okay, good. Yeah, so I'm Abby Fahim, as she said. I'm assistant professor of ophthalmology at University of Michigan. I see patients with inherited retinal diseases, and I have a research program investigating dysfunction of retinal pigment epithelial cells, which you see up here, in inherited retinal diseases, including choroideremia. I'm very grateful to have the support of the Choroideremia Research Foundation for my work, and I'm going to give you an overview of what we've been doing in my lab to understand this disease better so we can work towards a treatment. So choroideremia, as many of you know, it's an X-linked recessive chorioretinal dystrophy, which involves degeneration of the retina, the choroid, which is a blood supply for the retina, and the retinal pigment epithelium, or RPE, which is a supportive layer of cells that sits between the retina and the choroid. And in particular, the early and dramatic loss of the choroid is really what sets choroideremia apart from many of the other inherited retinal dystrophies. And the onset is in childhood or adolescence, often with legal blindness in early adulthood. So to further make this point, I want to present to you this clinical contrast between choroideremia and other inherited retinal diseases, and it really raises some questions about the choroid and retinal disease. So what I'm showing here is two patients with retinitis pigmentosa, and this is the most common inherited retinal disease, and they both have mutations in a gene called USH2A, and the top patient has what I would call moderate RP, and the bottom patient has severe RP. And the severe patient is legally blind and has choroidal atrophy. So what I'm showing here on the right, these are OCT pictures, and they show a cross section of the retina. And the lacy network that you see under the retina, that's the choroid. And it supplies nutrition to the retina and the retinal pigment epithelial cells. And in this patient here, the choroid is about the same thickness as the retina, and that's normal. In fact, it's hard to make out the bottom of the choroid, but it's about here. And so the choroidal thickness is about the same as the retina. But in this bottom severe patient, the choroid has become quite thin, and there's choroidal atrophy, which means loss of choroidal tissue. And that's because choroidal atrophy like this is quite common in advanced retinitis pigmentosa after there's been extensive damage to the retina. And the choroidal atrophy is likely either due to inflammation that gets incited by retina cell death, or loss of growth factors from the retina cells, or some combination thereof. And so now that you've seen that, now this is choroideremia, and this also has choroidal atrophy, but it's quite different. So you can see on the OCT that the choroid is thicker in the center, and that corresponds to this orange colored part of the photo where there's still preservation of the choroid. But then as you go towards the edges of the OCT, this choroid part gets very, very thin and completely disappears here, much more so than in the previous slide. And this corresponds to the whiter part of the photo that looks kind of white. And that's where it really is no choroid. Both the choroid and the retinal pigment epithelium are gone in this area. And it's pigment cells in the choroid and retinal pigment epithelium that give the retina its nice orange pigment. And so that's why it's white in this area. And so that's why it was called choroideremia, because that's the most striking feature when you look in the eye, is that the choroid is gone. But interestingly, most people in the field today suspect that the primary pathology actually starts in the retinal pigment epithelial cells, and that it's really dysfunction of the RPE cells that ultimately lead to photoreceptor and choroidal death. And it's fairly easy to understand how choroidal death could then contribute to RPE and photoreceptor death, since the choroid supplies the blood for these tissues. So all of this leads us to ask, how does RPE dysfunction in choroideremia lead to choroidal death? So the RPE is uniquely positioned to impact both the photoreceptors and the choroid. This is intuitive because the RPE sits between these two other tissues, which line the back of the inside of your eye. And this figure shows us the layer of RPE cells sandwiched between the photoreceptors on this side and the choroid on this side. So in this figure, the front of the eye would be towards the bottom of the page, and the back of the eye would be towards the top of the page. And this is a zoomed in diagram of what's lining the back of the inside of your eye. The photoreceptors are the light sensing cells, the rods and the cones that sense light and send the image to your brain, and have all these little processes called outer segments that intercalate with the processes in the RPE cells called microvilli. And the RPE cells have a very close relationship with the photoreceptors. And we know a lot about that relationship. The RPE supports the photoreceptors in numerous ways, including secretion of growth factors, phagocytosis, which is a process in which RPE cells eat and digest the old used up fragments of outer segments that the photoreceptors don't want anymore. And the RPE partners with the photoreceptors to carry out the visual cycle, which is a cycle of biochemical reactions that allows your eye to sense light. And there are more functions as well, but we know far less about the relationship between the RPE and the choroid. How do RPE cells support the choroid, or do they support the choroid? We think of the choroid as supplying nutrients and taking away waste, so the choroid is clearly important for the survival of RPE and photoreceptor cells, but is the inverse true? Are RPE cells important for the survival of the choroid? Or put another way, can dysfunction of RPE cells cause choroidal death? So there have been a few reports of findings in donated eyes from affected males and affected female carriers with choroideremia, and they found that there were widespread irregularities in thickness and pigmentation of RPE cells, even in areas of normal photoreceptors. So you can see how lumpy bumpy these RPE cells are. Some parts are very thick, some parts are very thin, as compared to this age-matched control who has a nice, normal, even layer of uniformly sized RPE cells. So this suggests that RPE cells are primarily affected in choroideremia, meaning that the choroideremia mutation leads to disease in the RPE cells specifically, which then go on to impact the photoreceptors and choroidal blood vessels. So we know that the gene mutated in choroideremia is called CHM. This gene encodes for a protein called REP1, or Rab escort protein 1. So the REP protein in this diagram is the large purple shape, and its job is to attach fatty chains called prenyl groups to Rab proteins. The fatty prenyl groups are shown as these black squiggles, and the Rab proteins are these small ovals. So Rabs are these small proteins that direct traffic in the cell. So one of the primary ways that proteins get transportation to different parts of the cell is by hitching a ride in vesicles made of lipids or small fat molecules. And these fatty bubbles carry around proteins to different parts of the cell, including the surface of the cell, to be secreted to the outside of the cell. So you can think of these vesicles as like the bus system of the cell, and then the Rab proteins would be the bus drivers that direct the vesicles where to go. And the way Rab proteins attach to these vesicles is by having these fatty prenyl side chains that insert into the lipid vesicles. And the Rep protein in purple over here is required to put those prenyl side chains onto Rab proteins. So without the Rep, the bus drivers can't find the buses, basically. So what I've tried to show you so far is that one, choroideremia has an early and dramatic loss of choroidal blood vessels that we can't yet explain. Two, the problem is likely to start in retinal pigment epithelial cells that sit right next to the choroid and the photoreceptors. And three, the genetic defect in choroideremia is likely to lead to errors in directing traffic of proteins. So what could the RPE cells be doing or not doing in terms of protein traffic that could be damaging the choroid? So we know that RPE cells are prolific secretors of growth factors in a polarized and directional fashion, meaning that some proteins are secreted much more towards the apical surface, which faces the photoreceptors, and some proteins are secreted much more towards the basolateral surface, which faces the choroid. As an example, RPE cells secrete something called PEDF, a factor that inhibits blood vessel growth towards the photoreceptors, and VEGF, which promotes blood vessel formation towards the choroid. So these two growth factors have opposite effects and are secreted in opposite directions. So you can start to imagine that upsetting this balance of polarized growth factor secretion could really have a big impact on how the RPE cells are able to support or potentially damage the surrounding tissues. So our hypothesis is that there's altered directional protein secretion such that the balance of growth and inhibitory factors secreted towards the choroid is skewed towards inhibition and choroidal death. The field of regenerative medicine has been growing, and we can now use pluripotent stem cells, in our case, induced pluripotent stem cells, in which blood cells or skin cells are induced to become stem cells by reprogramming them to a pluripotent state. Pluripotent meaning that they have the potential to become any cell in the body. And we can direct differentiation programs to generate different cell types. And in particular, in the field of retina, which has really led the way in regenerative medicine, we can generate retinal organoids that recapitulate human development in the various cells of the retina, including photoreceptors. And we can also generate excellent retinal pigment epithelial cells. So to study RPE dysfunction in choroideremia, we use RPE differentiated from induced pluripotent stem cells. The line that we're using is derived from adult skin fibroblasts. RPE is a default differentiation pathway if you deprive stem cells of fibroblast growth factor. By about day 40, we start to see islands of pigmented RPE. We pick those islands with a needle at a microscope, and then we plate them onto plastic. And after a few weeks, we then move them over and plate them onto these membrane supports that are suspended in media. And that allows us to separate the apical surface of the cells from the basolateral surface. And I'm going to show that to you in a minute. So one of the things we're doing in our lab is to characterize the RPE polarized secretome towards the apical and basolateral surfaces. So shown in this diagram, this is an RPE cell again. So this is again the apical surface with microvilli that envelop the photoreceptor outer segments. Numerous growth factors and other types of proteins are secreted from the apical surface towards the photoreceptors. And then this down here is the basolateral surface with these infoldings that increase the surface area. And numerous proteins are secreted from the basolateral surface towards the choroid. Now, when scientists use the suffix ome, O-M-E, they're generally referring to a complete set. So it's a way of looking at an entire set of genes, if you say genome, or proteins, if you say proteome, or RNA transcripts, as in transcriptome. So it's a way of looking at the complete picture without biasing your search to just a few proteins of interest. So when we say RPE secretome, we're referring to the entire set of proteins that get secreted from RPE cells. And when we say polarized secretome, we're referring to the fact that we care about which way the proteins are being secreted. We don't just want to know what's secreted, but also what direction. So several studies have looked at RPE secreted proteins with the RPE growing on a traditional plastic well, shown here. And when the RPE cells are on plastic, they polarize with the apical side towards the media and the basolateral side just sitting on the plastic. So when you investigate secreted proteins, you collect the media to see what proteins are there that were secreted from the cells. So when you look at the secretome of RPE sitting on plastic, you're really just looking at what's being secreted from the apical surface of the cells, because only the apical surface is exposed to the media. And it really doesn't capture the basolateral secreted proteins to which the choroid would be exposed. So we grow RPE on transwell filters, which separate the apical and basolateral chambers. So you can collect the media separately from each chamber to investigate the apical secretome and the basolateral secretome. And media is collected for protein analysis. We do this with the proteomics facility here at the University of Michigan. And this is just using normal control RPE. We see that the overall trend of the RPE secretome is that the majority of proteins are secreted apically more than basolaterally. So this is called a volcano plot. It shows all proteins that had at least twofold difference between apical and basolateral chambers. The red dots were more apical, the blue were more basolateral. So you can see that more proteins were skewed towards apical secretion compared to basolateral. And pathway analysis showed that many biological processes, including protein transport, protein localization, and various metabolic processes were significantly enriched in the differentially secreted proteins. Some of the proteins had known effects on blood vessel growth, including VEGF or vascular endothelial growth factor, and PEDF or pigment epithelium derived factor, and also complement factor H, which is it has a known genetic association with age-related macular degeneration. So we want to use these same types of studies to investigate protein secretion in choroideremia and how that's altered compared to control. So to do this, we used CRISPR gene editing technology in our stem cells. I'm sure many of you have heard of CRISPR. It's been in the media a lot. It's sometimes described as genetic scissors because it's a method of editing specific genes. And much of the disease modeling literature using stem cells uses patient-derived induced pluripotent stem cells, which involves taking a blood sample from a patient who has the disease you're interested in, like choroideremia, and making stem cells that you can then use to generate retina cells that have the choroideremia mutation. And then for any experiment with a disease model, you need a normal control. So in this situation where you use a patient sample as your disease model, you would then use samples from an unrelated normal individual or an unaffected sibling to be the control. And that approach does have some advantages. You're using cells from a patient with known disease. They have not only the particular mutation you want, but they also have a particular genetic background that has manifested the disease. However, for a genetic disease like choroideremia that has 100% penetrance, that means a disease-causing mutation in choroideremia in the CHM gene always leads to disease. You don't need to worry as much about the genetic background. So actually, we can knock out the CHM gene in any stem cell line. It doesn't have to be from a patient. And when we start with a normal stem cell line and knock out the CHM gene, this has an important advantage that we can now use the non-modified original stem cell line as the control. And it's exactly the same genetically as our disease line, except for the CHM gene knockout. It's what we call an isogenic line, where the two lines are genetically identical, except for the missing CHM gene. So this was the approach that we took using CRISPR-Cas9 to knock out the CHM gene. And you can see here, this is called a Western blot, where this dark band is looking at the REC1 protein. And you can see that our knockout line doesn't have any REC1, whereas our control line has REC1. And also this is another type of RPE cell line that has the REC1, but our knockout line does not. So we've made RPE cells from these stem cells and have done secretome experiments with the choroideremia RPE. And we have some potentially exciting results, which are too early to share, as we really need to validate what we found and do some additional experiments to see if the differences in secreted proteins have biologic relevance. But the goal of this work is to understand how the RPE may be negatively impacting the choroid. And we're also very interested in what the RPE is secreting towards the photoreceptors and how that changes in choroideremia. But our initial focus right now is on the choroid based on the unique choroidal atrophy in choroideremia and our interest in investigating why that happens. So another aspect of the cells that we're interested in is the tight junctions. And this diagram shows tight junctions. So these are two cells side by side, and the tight junctions are at the top of the cells, and they connect the cells together. So they're made up of a collection of proteins, and it creates a sort of a bond between the cells that makes it really hard for water and other ions to pass through, but it's selectively permeable to certain electrolytes, depending on the composition of the tight junction. So tight junctions are dynamic structures. It means they don't just sit there. The components of the tight junction are recycled on an almost continuous basis. So component proteins are leaving the tight junctions and coming back to the tight junctions all the time. So you can imagine, depending on the rate of how fast those building blocks are leaving and coming back, the tight junctions can be tighter or looser at different times. And in fact, there's some evidence that there's kind of a diurnal rhythm to that in terms of how tight or loose the tight junctions are. So we can measure how tight the tight junctions are at any given time by measuring what's called the transepithelial electrical resistance. So when we plate the cells on this permeable support and it's suspended in media, like I said before, the apical chamber is separated from the basolateral chamber. And we can measure the electrical resistance by using these electrodes that straddle the wall of the chamber. And so one electrode's in the apical side and one's in the basolateral side. And what we're measuring is resistance to the flow of current across that membrane. And a lot of things contribute to the resistance, the membrane itself, the cells themselves, and where the most current flows through is between the cells at the cell-cell junctions, where the tight junctions are. And so it's the strength of the tight junctions that determines how much current gets through and how high the resistance is in one well of cells compared to another. And this is just some pictures of our RPE cells staining for different tight junction proteins. And you can see how nicely the cells are outlined because these tight junction proteins are all along the cell-cell junctions. So the reason I'm interested in tight junctions in choroideremia is because tight junctions are regulated by Rab proteins. And Rab proteins are, of course, at the center of the biochemical defect in choroideremia, because that's the whole defect, is that Rab proteins don't get the prenyl groups added. Rab13 in particular is known to have a major role in regulating tight junctions. And we have found in our choroideremia RPE cells that the tight junctions respond differently to stress than control cells. So we have this gene mutation in REP1, which regulates the function of Rab proteins. We know that some Rabs are important in tight junction function, and we have choroideremia RPE in our lab with altered tight junction behavior. So now we're investigating whether Rab13 and other Rabs may be playing a role. Now the question is, how do alterations in tight junctions, how would that lead to the choroideremia disease? And that's not clear yet. One thing we know from the literature is that tight junctions may be a hotspot for protein secretion from the cell. So proteins don't necessarily get secreted just at random all along the cell surface. There's evidence that some of the protein secretion from the cell is targeted specifically to this location of the cell surface where the tight junctions are located. And there's also evidence that, like I said before, tight junction tightness varies according to a circadian rhythm, and the tight junctions become looser at certain times of day. And this might be important for the circadian rhythms and the light cycles of RPE cells. So we're continuing to investigate both protein secretion and tight junction composition and behavior in our cells. And our goal is to identify disease processes that can be specifically targeted for intervention to alter the course of disease. So I'd like to talk about some of the other potential applications of stem cells in inherited retinal diseases. My work thus far is focused on using stem cell derived RPE to model choroideremia. But as many of you know, you can also make photoreceptors or rods and cones from stem cells. And this slide is showing retinal organoids, which are these tiny retinas growing in a dish. And here we are isolating these organoids with a needle under a microscope. And they differentiate and develop along a timeline that's remarkably similar to human fetal development of the retina, meaning it takes at least 4 months to start seeing rudimentary photoreceptors with outer segments in the organoids, and at least 6 months to see more developed outer segments. And stem cell derived eye tissues, including both photoreceptors and RPE cells, can be used for either disease modeling or regenerative therapies. So what I showed you with my research is an example of disease modeling, in which we grow cells that have the disease of interest in one sense or another, whether that's by genetic manipulation or inducing disease some other way. And we do that in order to understand the disease and develop new treatments. Regenerative uses of stem cells would be using the stem cells to grow healthy photoreceptors or RPE that would be suitable for transplantation into a patient to replace cells that have been lost from retinal degeneration, whether that's due to choroideremia or macular degeneration or infection or really any cause, anything that results in retina cell death. So I want to show you this really cool example of disease modeling using retinal organoids. On the left, we see photos of patients with an inherited retinal disease called retinoschisis. And in this disease, there's a mutation in a gene called retinoschisin, which is a protein in the retina that holds the retina cells together. It's important for cell-cell adhesion. So when this protein is defective, you get a separation of the layers of the retina. So these are OCT pictures showing cross sections of the retina. And you see all these black spaces where the retina layers have separated from each other. And so these authors made retinal organoids with a retinoschisis mutation, and look at all these spaces that have formed within the retina in the organoid. So it really closely mimics what we see on the OCT. It's just a remarkable model that they've created. So in terms of the therapeutic potential of stem cell derived photoreceptors, this is late breaking news from our Kellogg visiting professor, Robin Ali, who's at King's College London. And he just published this paper a few months ago in Cell Reports. For a while now, Robin Ali and many others have demonstrated differentiation of human photoreceptors from pluripotent stem cells. And Robin in particular has led the way in demonstrating enriched populations of cones. And David Gam at Wisconsin has demonstrated function of retinal organoid photoreceptors in terms of their response to light. This paper that recently came out really shows that transplantation of stem cell derived cones into a mouse with retinal degeneration leads to integration of the transplanted cells with evidence of synaptic connections and evidence in the mice of improved retinal function and visual behavior. So we've really come a long way in terms of having a strong preclinical rationale for trials in human patients. And we're all waiting to see when we'll have a phase 1 trial for this type of technology. So in conclusion, in choroideremia, we have death of photoreceptors, RPE cells, and choroidal blood vessels. In particular, the early loss of choroidal blood vessels is unique among inherited retinal diseases. There's evidence that the RPE cells are dysfunctional in this disease, and this dysfunction may be detrimental to the surrounding tissues and may be contributing to the death of the photoreceptors in choroid. And loss of the choroidal blood vessels in turn can clearly contribute to the death of the RPE and photoreceptors, which rely on the choroid for nutrition. The genetic defect is in a protein needed for prenylation of Rab proteins, which are important for trafficking and secretion of proteins from the cell and for regulating tight junctions. We're actively investigating the proteins secreted from the choroideremia RPE, in particular to see if there are changes in protein secretion towards the choroid that would be detrimental to the health of the choroidal blood vessels over time. And our goal is to identify pathways and proteins that we can target in future small molecule screens for novel therapeutic intervention in this disease. So I would like to acknowledge all my lab members, my mentors, our leadership at Kellogg, and of course, my funding from the Choroideremia Research Foundation. And I also have a K08 from the National Eye Institute and a previous grant from Eversight. So thank you so much for listening. I'd be happy to take any questions.
Well done. That was super. Thank you so much. I just have to share with everybody when Dr. Fahim and I were talking earlier in the week and swapping email, I was doing a little bit of reading on what the grant, her write-up for the grant was and the application. And I thought, oh my goodness, how can we make this be understandable? And I'm telling you, we right now have one of the largest groups that we've ever had, and you did phenomenally.
Okay.
It was a great explanation. So thank you so much.
You're so welcome. I hope that that was accessible and somewhat interesting for people.
No, it really was. And right now it looks like we have a couple questions coming in. So just take a few minutes, folks, and type your questions and I'll ask them to Dr. Fahim. This is really, really cool. And there's a lot of people that may have heard of this. I didn't, but as I was taking notes, I'm just going to turn my notes over. The tight junctions, that was a new concept to me. And
Yeah.
it was super exciting how you were able to connect the dots for us on how that may be connecting with the Rab proteins, and then transplantation, it sounds like, is the goal. And I'm looking forward to actually watching this again. We could use some excitement. I think our organization could use some positive things. And when you say this is exciting, and perhaps down the road, the transplantation concept might be sooner. We of course love David Gam. He's if we had a baseball team, he'd probably be one of the pitchers. So we're super glad you're on our bench too. Here, let me give you some of these comments that are coming in. So Brent Pike, who's always on these calls. Hello, Brent. Remarkable explanation of how RAB13 and other elements interact. Thanks for making it easy to understand. Keith says, excellent talk and elegant research. Thank you for both. Is there any evidence or do you have any thoughts on the possible impact of RPE stem cell transplant could possibly have on restoring choroidal function?
Yeah, that's a really great question. You know, I think in theory, if you were to do an RPE transplant early, in theory, perhaps you could halt or slow some of this. I think ultimately the best transplantation strategies for this and many other diseases is going to be a bilayer or trilayer transplant. In other words, you know, as I mentioned, really the problem is degeneration of 3 types of tissue: the photoreceptors, RPE, and choroid. And I think to really restore vision in those blind spots where vision has been lost, you'd have to replace all 3. And there are people working on that, doing these scaffolds with co-culture of the different cell types. At first, you know, the first step is kind of doing the photoreceptors and RPE together, but people are also working on then adding the choroidal endothelial cells for a tri-layer type of implant. But you know, that will be further down the road in terms of clinical application, but I think that's definitely within reach. And yeah, but to sort of get back to the specific question of whether you could just transplant RPE and sort of rescue the choroid, I suppose you could in theory. You'd have to do it pretty early. And so, you know, I guess those types of preclinical experiments haven't been done yet. I mean, part of the problem is that our mouse model, I say our, it's not mine, but in the field, the mouse model of choroideremia is a little confusing. It's difficult to breed because it's lethal to the male mice in utero. And so basically you have to use a very complicated breeding scheme to generate the choroideremia mice. And from the publications about it, they don't mention anything about choroidal atrophy. The mice do have a retinal degeneration, but they don't have the choroidal atrophy that's so central to the human disease. So it makes some of the preclinical work challenging. Sorry, that was a long-winded answer.
No, that was a good answer because you answered the next question. You didn't even know it. Karen was asking, is there a mouse model for choroideremia?
Yeah, there is. It's so as I said, the males don't survive because the choroideremia gene is thought to be important in the placenta for mice. And you know, of course, the mice only have the males only have the 1X chromosome. In female mice, it's lethal if the choroideremia mutation is inherited from the mother, but not the father. And so that's where it gets into this complicated breeding scheme of trying to generate the mice. But they've done some work doing tissue-specific knockouts in RPE only and photoreceptors only, and there's a lot of papers out there about it. It's a good model in some respects, and in other respects doesn't totally recapitulate the human disease.
Let's see. Do you think that a different animal model would prove effective if you had such as a dog or a porcine model?
Yeah, potentially. Yeah, that could potentially be good. You know, I know there's a zebrafish model, but having a larger animal model could be helpful. There are some unique things about the mouse placenta that I think may not be true for the larger mammals, so that it might be easier in terms of, you know, breeding them and having them survive. And who knows, we might get a better a better model of the human disease with the choroidal atrophy and everything.
That's it in terms of questions. And let's see, here we go. What other researchers are doing work that you follow and admire? That's from Kathy, our executive director.
Yeah, oh, that's a great question. Well, so I mean, a lot of the work in choroideremia, as you guys probably know, has been done by Ciabra, who did the mouse model and has done a lot of the prenylation experiments and then in terms of stem cell work and disease modeling, David Gam is excellent, and Bud Tucker at Iowa has done a lot of disease modeling, and so yeah, a lot of people.
One of the things that we're trying to do too is ask our researchers if there's just one or two things about you personally that you could share with our group, kind of strengthen that relationship, or what would you like to share something about you or your family?
Sure. Well, yeah, my husband's a neurosurgeon. There you go. There's something. And we live here in Michigan, and we have 4 kids ranging from 15 years to 16 months. So a widespread. Yeah, and we're doing well. we're taking it day by day.
Yeah, I would think you'd have to with that spread.
Yeah.
Super good.
Yeah, I'm very grateful for the opportunities I've had, and everything's going well so far. So.
I just got a text message. Thank you for sharing that, by the way. That's pretty neat. Thank you. We had Eric Hartman's asking a question, or has a comment here. I've just got a text message. Probably is one of the best explanations I've heard of the REP1. Could allogenic transplantation, I hope I'm not butchering that word, allogenic transplantation of healthy RPE be accomplished in areas where there are CHM RPE? Meaning, could those tight junctions be adjusted to allow for the insertion of healthy RPE that could hook up where those tight junctions are?
Yeah, that's a really interesting question. So certainly clinically, there have been, not in choroideremia patients, but in macular degeneration patients, Stargardt patients, which is another inherited retinal disease, there have been some clinical trials already with stem cell derived RPE transplantation, some phase 1 safety studies. And, so far, RPE has really been transplanted in areas where the RPE has already been lost. So there really isn't anything there. And then you put the RPE in, and it kind of takes over the area. If you still have, viable RPE there and you transplant more RPE, I really don't know, because RPE is meant to form a monolayer. And it really functions well as a monolayer. It doesn't function well as cells heaped on top of each other. So, I guess we haven't tried that, but I would be somewhat concerned that maybe there would be too many cells in one spot, that it wouldn't function well as a nice monolayer. So I don't know, I guess, is the answer. But I think transplantation therapy works best in an area where there's been cell death and things have been lost. And if you still have viable cells there, ideally you would improve the function of those cells, either through gene therapy or some small molecule therapy that improves the function of the cells. I think it's all speculative, right?
Right. It's all research.
Yeah.
But it's exciting. I think it's more exciting for the folks that are more advanced with their CHM, with more vision loss. This provides hope. And one of the things we try to do as a foundation in our mission and is to educate and to support, but a lot of that is providing hope and keeping our eyes lifted and in hope that something is coming. So this definitely hits the spot.
Yeah, so I think multiple approaches are important. I think the transplant therapy is really important for those later stages where you have to replace what's been lost. And, one of my goals is to be able to do small molecule screens to improve the function of the RPE. So, to either improve protein transport processes or improve prenylation. That's really the goal of my research right now, is to find what pathway or protein am I trying to target, to really target with some type of small molecule therapy potentially.
So next steps would be what in an effort? No more questions. I think we'll just finish it up with that. Next steps, and what would be the next partnership that you would like to see?
Yeah. So, we're trying to validate a lot of the results that we've gotten from proteomics to see what are the most important differences in protein secretion that we're seeing, and then test the effects of those proteins in potentially a co-culture system with choroidal endothelial cells. We'd also like to see what are the most underprenylated RABs in Choroideremia RPE. All the prenylation experiments with RAB proteins in Choroideremia have not been done with RPE cells. They've been done with other types of cells. And, Choroideremia is an eye-specific phenotype. So we really want to use ocular cells, specifically RPE cells, to look at these questions. So, yeah.
Super good stuff. That's awesome. For a Friday, this is great news. It's a good way to end. It's a good way to end the week with all of your good work. And please extend our gratitude to your team. It's always great to see a picture. Thank you so much. And again, more people are chiming in. Thank you, Dr. Fahim. It's always great information. So we look forward to good things, hearing more things, and reading what you guys publish.
Thank you. It's so great to hear all these comments. I'm glad you guys enjoyed it.
Very much so. Well, thank you everybody for coming. We'll be having another session next month with another researcher. So we look forward to seeing you all then, and have a great weekend. Bye.
Bye.